The ancients called it the deadly carrot because their thirsty camels would come off the caravan trail, eat it, and die. The Roman Emperor Nero mixed it with frankincense to treat bruises. Up until the early 20th century, it was used as a plaster to treat rheumatism, a plaster that caused severe side effects. The Thapsia garganica plant, a weed that grows abundantly in the Mediterranean, kills by disrupting the calcium balance in cells, all cells. Now GenSpera has targeted its active ingredient, thapsigargan, to kill only the cells we select, cancer.
We have taken the powerful toxin thapsigargan and found a way to keep it inactive until it finds the cells that it's been programmed to seek. Then it releases its active ingredient and destroys all of the targeted cells.
In the case of GenSpera's first drug, G202, this target is PSMA, an enzyme which is only found on the walls of the blood vessels that feed cancer tumors, regardless of the type of cancer. Unlike anti-angiogenesis drugs, which prevent the formation of new vessels, G202 also destroys existing cancer blood vessels, not just the ones that are forming. G202 is currently in phase 2 trials at multiple cancer centers in hepatocellular carcinoma, or liver cancer, as well as in the brain cancer glioblastoma. GenSpera also plans to initiate G202 trials in prostate cancer and renal cell carcinoma in 2014. GenSpera's active ingredient has several unique cancer-killing properties. It kills all cancer cells regardless of their rate of division, and it kills cancer stem cells as well because it will kill any cell at any time unless directed. GenSpera directs it.
We deliver this powerful cancer killer via a prodrug platform that renders the drug inactive until it encounters our selected target. We achieve this by attaching a peptide to the molecule in a form that we've derivatized specifically for that purpose.
The peptide acts like a pin on a grenade. A grenade is safe until you activate it by pulling the pin. Like a molecular grenade, G202 circulates benignly in the bloodstream until it encounters the enzyme we've targeted, which is found only on the blood vessels that feed cancer tumors. The grenade pin targets these cells, which then pull the pin. As the grenade activates, the active ingredient is released and falls into the cell. This happens again and again with each passing grenade until enough of the drug accumulates inside the cell to block its calcium channels. The cell dies. All the blood vessel cells die. The tumor is starved and dies as well.
The beauty of this prodrug platform is that it is completely modular, and we can select different targets depending upon the particular cancers that we are going after.
Since G202 circulates harmlessly throughout the body until it finds the enzyme it's seeking, it won't affect any other cells in the body, potentially avoiding some of the side effects of standard chemotherapies. If successful, G202 could treat multiple cancers with fewer side effects than today's therapies.
I've had a long career in cancer drug discovery, both at major biopharmaceuticals as well as cancer nonprofits, and I've never seen a drug with this potential. Most cancer drugs we call effective, meaning they make tumors go away, but they almost inevitably come back, leading to an increase in median survival of only a few months. We want to change the cancer vocabulary from effective to curative, and we believe that our drugs have the ability to make that change.
Thapsia garganica has been around for centuries, but it took a group of scientists to explore the properties of thapsigargan and combine it with modern biochemistry to create a powerful cytotoxin that kills on demand and in a targeted fashion. At the University of Copenhagen in Denmark, Dr. Søren Christensen, a natural products chemist, has been studying the pharmacology of Thapsia garganica since the 1970s. often vacationing with his family and other scientists in regions where the plant grows to pick its toxic seeds.
Chemical and pharmacological investigation of natural products have led to several scientific breakthroughs. The Mediterranean plant Sapsia garganica has been used for centuries in traditional European and Arabian folk medicine, but nobody knew why it worked. We became interested in unlocking the chemistry of this plant and to find out how it worked inside the body.
While Dr. Christensen was studying the Sapsia garganica plant and isolating its active ingredient, we were working on trying to figure out ways to trick cells into undergoing apoptosis or programmed cell death. And so we were looking for agents which could do that. Working with Dr. Christensen, we discovered that thapsigargan causes an elevation in the calcium levels within all cells. And the result of this is that it activates this apoptotic or programmed cell death pathway, so it kills all cells. The trick was to modify thapsigargan so that the enzyme peptide grenade pin could be used to activate it selectively.
Dr. Samuel Denmead, together with Dr. Hans Lilja, designed the first peptides that could be attached to an analog of thapsigargan and then removed by the target enzyme at the site.
The use of thapsigargan as a drug requires a large and sustainable supply of the compound. Our team is working on metabolic engineering of mast cells to make them produce thapsigargan. Since mast cells can't be grown in large vats, these cells will become a sustainable supply of the compound.
The MOS project is one part of our global strategy for drug supply. It will give us vast quantities of thapsigargan. Importantly, it will give us a year-round harvest that is literally cheaper than growing weeds. We are focusing one of nature's most powerful toxins on cancer for the very first time. Once we discovered how to turn it into a drug that was safe for normal cells, we were awed by its potential to treat one of mankind's deadliest diseases. That's why our name means Genesis and hope. At GenSpera, we are creating therapies and generating hope.